10.3 DNA replication. So the structure of DNA holds the secret of replication. Base pairing provides a perfect copying mechanism. If we look here, we can see that we have our two bases, guanine and cytosine, that will pair up nicely in hydrogen bond, making 3 hydrogen pairs here. So this is one set of complementary bases. On the other side, we have adenine and thymine, which will form 2 hydrogen bonds, and this forms a perfect complementary base pair with each other. So by base pairing, we have a very eloquent way of copying DNA. So during replication, the hydrogen bonds are unzipped by an enzyme called helicase. This area which is being unzipped is called the replication fork. Each of the 2 original strands serve as a template for construction of a new matching strand. So the first thing that we need to do is we need to unwind the DNA. That's done with the enzyme helicase. The hydrogen bonds between the complementary base pairs, if we look at our picture here, they kind of split apart. And now we have free-floating complementary base pairs, or free-floating nucleotides, which can come in complementary base pair, form those hydrogen bonds that we talked about with their partner to give us 2 identical strands of DNA. This process is called semi-conservative replication, and we can see here we've got our parent strand, our parental, which is this purple. It unzips, and then one goes to one side, one goes to the other as the template, and then we've synthesized our new strand. We can see then on the next cell division that we end up still with our original strand here. It never disappears. It will keep being used over and over and over again. When we replicate DNA, bases can only be added in one direction. Therefore, one polymerase moves in the same direction as the replication fork, and that's called the leading strand. And the other one moves in the opposite direction away from the helicase. That is referred to as the lagging strand. So in this picture you can see we have the helicase, which is the enzyme here that is unzipping. It's going to go through and unzip our hydrogen bonds so that we can readily complementary base pair our nucleotides, which we did in our exercise today with our Kinnex molecules. In this picture of the DNA molecule being replicated, you can see here is our helicase that's unzipping our complementary base pairs, and then we have our leading strand, which is able to move in the same direction as our helicase. Now, because the opposite, remember our sugar phosphate backbones, remember they're moving in opposite directions as each other. So in order to replicate DNA on the opposite strand, we can only do it in little tiny sections. So we'll make this section, and then we'll go back, and then we'll make the next one, and then we'll keep going. And these little fragments are referred to as our Okazaki fragments that we're going to make. Now this is referred to as our lagging strand because it takes longer to copy this strand right here than it does the leading strand. So here is just a summary of DNA replication. So again, it's showing you here that the first thing we do is we're going to unwind the DNA with the helicase. That's the enzyme that unzips our DNA. This picture goes in a little more details in that we have little proteins that's going to keep the original strands from coming back together. So it's going to keep it apart so we can replicate it. And then we have our leading strand here, which is moving in the same direction as our helicase, so it can move along promptly and keep up with the replication fork. Now on the bottom here we have the lagging strand, and remember we're gonna synthesize this in little tiny pieces because it's moving in the opposite direction. It's moving in this direction versus this direction in our replication fork is moving in that direction. So this bottom strand, that lagging strand, takes longer to replicate than the leading strand. With prokaryotic replication, prokaryotes only have one circular strand of DNA. So what we'll find is that we have this origin where we start, and then we'll just have two replication forks, one moving this way around the circle and this one moving around the other side of the circle, until eventually they meet up, and now we have two strands of DNA, one for each bacterial cell as they divide. And when we talk about the rate of replication, it would take 53 days to replicate the largest chromosome in a human. And we know that we have 46 chromosomes, so that would take a long time to copy our DNA in order to make a new cell. So to speed up this process, we're going to use mini replication forks. For example, in the fruit fly, it has been known that at any given time, when a cell is dividing, that particular cell will have 3,500 replication forks going at once in order to get that fly's DNA copied in time for that cell to divide. Now sometimes there are errors in the replication process, as we noticed when you guys were copying your DNA molecules using the Kinnex models. But the DNA polymerase does have the ability to proofread. The problem is it can proofread it as it's putting the bases together. It can tell if there's a complementary base pair that's not aligned correctly. But if that particular mistake goes on into what we call gap 2, and we'll talk about the cell cycle in third quarter. So if it takes place later during the cell cycle, the DNA polymerase cannot tell which one is the parental strand and which one's the newly copied strand. So there's a 50-50 chance that the DNA polymerase will correct it in the correct fashion versus putting in the wrong complementary base pair. So this picture actually shows that dilemma. So right here we have G base paired with A, and if we had identified that mistake right away when the bases were put together, DNA polymerase would have no trouble recognizing which one belongs. Is it the G or is it the A? But like I said, in gap 2 we don't know, so there are two possible corrections here. We could either get rid of the G and put a T, or we can get rid of the A. We can get rid of the A if my pen will work, and put a C. Only one of those corrections will work properly. If it does the other one, we could lead to some kind of mutation. For our last slide, we're going to talk about mutations. There are different factors that can lead to DNA mutations. There are chemicals, UV light and other solar radiation. We talk about gamma radiation, X-rays. All of those things are from the sun, or they're, of course, man-made. When we talk about our X-rays, we don't want too many of those in a given year. Sometimes these mutations can lead to a positive or negative change in an organism, and we can have this included in our possible ways of making some genetic variation or some changes in genes. When a mutation causes mistakes in the cell cycle, though, that's frequently called cancer. And what happens then is the cell will then replicate over and over many times when it's not supposed to be undergoing cell division, and this leads to what most of us know what tumors are. And we'll talk about this more when we get into our genetics and cell bio fourth quarter.